like this. So it's my pleasure today to introduce David Knowles, who's our newest faculty member, arriving only 8 weeks ago.
Yeah, close to the 6, I think, yeah.
David graduated from the University of Cambridge with an honors degree in physics and engineering and received a master's degree from Imperial College in London in bioinformatics and systems biology. David went on to earn a PhD in engineering with a focus on machine learning from the University of Cambridge. And he joined the Genome Center from a postdoctoral position at Stanford University, working with professors Jonathan Pritchard and Sylvia Plevritis. David has performed with distinction at every level. For example, he's received an International Award for Innovation in Bayesian Statistics in recognition for distinction in mathematics at every level of his education. David's research focus is on the development of novel machine learning methods and their application to data analysis for genomics, particularly directed towards understanding genetic regulation of RNA transcription, particularly RNA splicing. David played a key role in the development of a program called Leafcutter, which is the most popular program now used to identify, quantify, and test variable intron splicing events. This work is fundamental to the goal of understanding the relationship between disease-associated DNA sequence variants and aberrant RNA splicing. This work is central to the understanding of disease mechanisms and the problem of understanding the consequence of mutations in the non-coding regions of the genome. This work is therefore central to a major effort at the Genome Center directed towards understanding the genetics of autism and other neuropsychiatric diseases. So David joins the New York Genome Center faculty with joint appointments in the departments of computer science and systems biology at Columbia. And we're very excited to have David here and look forward to his talk, as it says, Probabilistic Models of Transcriptome Dysregulation in Genetic Disease. David.
Thank you, Tom, for that very kind introduction. So I wanted to give a little sense of my background, but Tom's mostly already done that, but I'll give you a little bit more sort of detail on it. So when I was doing machine learning in grad school, it was a bit before this, this rise of deep learning. So the sorts of things we were working on back then and were excited about were, for example, Bayesian non-parametric models. The canonical example of this would be if you're fitting a mixture model or doing a cluster analysis and you'd like to learn from the data in a principled way the number of mixture components or clusters that would be appropriate. An example that I worked on in particular was doing this for factor analysis type models. So the model that you'd probably be the most familiar with in this setting would be principal components analysis. So how many principal components represent signal in the data rather than noise. I also worked on Bayesian non-parametric models for spatial temporal data, network analyses. So you think of, for example, gene networks or social networks, and also Bayesian hierarchical clustering. These big Bayesian models are challenging to fit to large-scale data. And one approach that's become popular is to use these methods called variational inference. And these offer deterministic alternatives to Markov chain Monte Carlo, which is the usual tool that Bayesian statisticians would throw at fitting these types of models. And the work I did here was to extend those methods to be applicable to a wider class of probabilistic models. And David Bly's group has actually really taken some of these ideas that we developed a few years back and really developed this into an active subfield in machine learning. During my postdoc, I got increasingly interested in, well, before that even actually in my PhD, I was involved in a number of collaborations in genomics. For example, looking at epigenetic changes during the development of cardiomyopathy, as well as looking at gene expression in the Mother Consortium in the UK, which was one of the first consortia to have large-scale observational gene expression data from many individuals in multiple tissues. And something that was really interesting from the genetic analyses point of view was that it's also a twin-based study. So there's a lot of genetic analyses that you could adapt from traditional twin studies to the gene expression setting. During my postdoc, I worked on various computational methods for genomics. Work I'm not going to really get a chance to talk about today is leveraging allele-specific expression to try to improve power to detect gene-by-environment interaction effects on the transcriptome. And a concrete example of that that was published last year with Yoav Gilad's group at Chicago was looking at the genetic basis of cardiac sensitivity to chemotherapy, so specifically doxorubicin. And that was using induced pluripotent stem cell-derived cardiomyocytes as a model for in vivo pathology. The work I'm going to talk about today, as Tom mentioned, revolves around splicing and alternative splicing. So, as you know, in higher eukaryotes like humans, the majority of genes are multi-exonic. And splicing is the process by which the introns need to be removed to generate mature messenger RNA. Alternative splicing is a phenomenon that there is variation in which regions are removed as intronic. So that variation can be across mRNA molecules within a single cell, between tissues, between species, between disease and health. And because different splicing choices result in different proteins, that can have different functional consequences. The example I like the most of this is the splicing of the tau protein. So tau is involved in stabilizing microtubules, which are important in pretty much all cells, but especially important in neurons, where the huge length of axons relative to a normal cell body size means that it's a huge challenge for these cells to transport the required proteins and nutrients up and down this enormous length. There are axons in your spinal column that can be multiple feet or up to a meter long. So that's obviously an enormous scale compared to the intracellular transport that you'd normally think of. So the microtubules form this effectively railway network that these proteins called kinesins can use to transport required proteins and other nutrients up and down the length of the axon. As you can imagine, if tau is dysregulated for some reason, then that can affect microtubule stability and affect whether the neurons are able to perform their duties appropriately. The tau gene is made up of 14 exons, which is relatively typical for human genes, so the average is around 9. And the splicing of exon 10 is particularly important. So there are two main isoforms of tau, 3R and 4R, and they are so named because if you have exon 10 included, then you end up with 4 microtubule binding domains, whereas if you don't, then you only get 3, because exon 10 encodes one of the microtubule binding domains. As you might imagine, if you have more microtubule binding domains, you end up with higher microtubule binding affinity. And what we observe in healthy development is actually that in fetal brain, you see almost exclusive expression of the 3R isoform, whereas in adult brain, you see very balanced expression of the 2 isoforms. So this is an example in normal healthy development where it appears there's a functional role for alternative splicing. But what we also see is that in many neurodegenerative diseases, including frontotemporal dementia with Parkinsonism, you see an imbalance of these 2 isoforms. In this case, increased expression of the 4R isoform. And in this case, at least, it's pretty clear that this is actually the cause of the disease because there are mutations that clearly drive this difference. So I'm going to talk about 2 projects that I worked on during my postdoc that are aiming to use RNA sequencing data to better understand splicing and particularly to understand the effects of genetic variation on splicing. So the first will be asking how we best quantify splicing from short-read RNA-seq. And the second project is looking at how we might predict the effects of variants or de novo mutations that we haven't observed before on splicing outcomes. So most methods for existing methods for quantifying splicing fall into two categories. The first I would call isoform ratio estimation methods. So I'm showing here an example of what this typically looks like, and this is a figure from a nice review paper by Leo Pachter in 2011. So here, for this gene, what Leo is showing is 3 different isoforms, so these 3 different colors, and corresponding RNA sequencing reads, and the ambiguity in where these reads could have come from. So this first read could have come from any of these 3, the second read from either the blue or the green isoform, and the only unambiguous read here is the spliced read D that must have come from this isoform. So there's a line of research trying to view the expression of each isoform as a hidden or latent variable, and then assign these reads to the isoforms. But this is, with short read data at least, a very statistically challenging problem. Especially for large human genes, this becomes close to unidentifiable. And that just means that the quantifications that you get at the end of the day are very noisy. And in practice, what this means is our power to detect associations between these quantifications of splicing outcome and covariates like genotype, is the power there is very low. Another approach is to calculate exonic percent spliced in. So the idea here is to concentrate on some focal exon. Assume for now the exons on either side are constitutive, i.e. they're always included in the transcript. And using reads that unambiguously support either inclusion or exclusion of that exon, and accounting for read and exon length, it's possible to get an unbiased estimate of the proportion of transcripts that included that exon. The problem with the exon centric view is that while it does a perfectly good job representing exon skipping, there are lots of other types of splicing events that we'd like to be able to quantify. One, for example, is 3' or 5' exon extension. There can also be cases where multiple exons are skipped at once. Another phenomenon we see is mutually exclusive exons, where either one exon or the other is included, but not both. And there can also be combinations of these different types of splicing events. So the approach that we took in developing Leafcutter was to focus on intron usage rates rather than exon usage rates. And while this is a simple change, it comes with a number of advantages. So firstly, introns are uniquely defined by spliced reads. If I can correctly align a spliced read, I know exactly what region was removed from the precursor messenger RNA. And then that also makes quantification easy, because to say how much an intron is being used, I can just look at the number of spliced reads that correspond to that intron. I'm also less reliant on having an accurate gene and isoform models, because the data itself is defining the units that I'm going to quantify. And it also lets us model more complex splicing events than if we were just looking at exon inclusion rates. So what does this look like in practice? So consider this gene with 4 exons. The black ones here are representing constitutively spliced exons, and then this cassette exon and a 5' extension on this 4th exon. The spliced RNA-seq reads might look something like this in this region. And what we still need is a unit against which we'll normalize. So in principle, you might think, well, we could just normalize relative to all the junction spanning or spliced reads for the gene, but that can work poorly if there's differences in what region of the gene is actually used. For example, if there's alternative TSS or alternative polyadenylation. So what we do instead is to look at splicing more locally, and we do that as follows. So we construct what we call an intron graph. So here, the splice sites correspond to nodes. So the splice sites are just the starts and ends of the introns. And we draw edges between nodes if there are spliced reads supporting that junction being used. The normalization that we then do is relative to connected components in this graph. So a connected component is defined by saying that any two nodes are in the same connected component if you can get from one to the other by traversing edges in the graph. So in this example, there'd be two connected components here, the first one here and the second one here. So then the quantification we use for leafcutter is the number of spliced reads for each intron relative to the number of spliced reads for the other introns in the same connected component. I'm showing again these different classes of splicing events that this lets you represent, as well as the spliced reads that would support each of these situations. So the first thing we wanted to be able to do with these quantifications was compare between conditions. So for example, you might want to compare between different tissue types. So this is data from GTEx comparing brain and heart RNA-seq samples for this RBFOX1, which is itself an important splicing factor. And what I'm showing here for this subregion of RBFOX1 is these, the thickness of these lines corresponds to is proportional to the number of reads supporting each of those junctions. And what I hope you can see intuitively here is that in brain, you're seeing greater usage of this first exon, whereas in heart, you're seeing greater usage of this second exon. So to actually test statistically for these sorts of differences, we use the counts themselves from the leafcutter pre-processing and feed those into a Dirichlet multinomial generalized linear model. I'm not going to go through the details of this, except to say that the multinomial likelihood means that we're looking at choice. So we're saying that the spliceosome is choosing which introns it's going to use out of a finite set within the component. The Dirichlet part lets us model additional noise on top of the multinomial sampling. And the generalized linear model is just convenient in that we can include whatever covariates we want. So those could be the one covariate here, for example, would be which tissue are we in, but you could also think about including technical covariates, so things like batch. The model fitting is done via STAN probabilistic programming language, which is actually developed at Columbia by Bob Carpenter and co. And it's a very nice tool for developing these sorts of models. And we can use likelihood ratio tests to get p-values and do hypothesis testing. So we initially applied this to the GTEx data set, which I expect almost everyone in the room is aware of, but this is a large NIH-funded RNA sequencing project doing RNA-seq in many tissues across hundreds of individuals, and has been a huge resource for the genetics and genomics community. So we've looked for differential intron usage across GTEx tissues. We find hundreds of differentially spliced introns. And what's really, I think, amazing about a lot of this signal is that we see conservation of those differences between tissues when we look in data sets where they've done RNA-seq in related species. So looking across mammals, we actually see up to, even as far as mouse, up to 8, depending on the tissue pairs, sort of between 5 and 10% of the differences that we see actually still being conserved, across hundreds of, you're getting up to like 100 million years of evolution at that point. And that is pretty amazing in that it suggests that a lot of those differences are actually functional because natural selection is maintaining them. Something that I like to show, partly because I think it's a nice advertisement for open science, is this paper looking at using RNA sequencing for diagnosing mitochondrial diseases. And what's cool about this is we put leafcutter and we put the code on GitHub and the paper on bioRxiv, mid-2016. And then only a few months later, this paper went up on bioRxiv and was apparently submitted to Nature Communications, using Leafcutter as part of their pipeline to look to do this diagnosis. So what they did was to take patient fibroblasts from 48 individuals who had exome sequencing but hadn't received any genetic diagnosis from the exome sequencing, and then use RNA sequencing to look for 3 different categories of gene dysregulation. So aberrant expression, where they found 1 event, and aberrant splicing using Leafcutter, where they were able to diagnose 5 individuals, and monoallelic expression, which allowed them to diagnose another 6. And they have some nice functional validation that I don't have time to go into detail on. One example that they found was a mitochondrial endopeptidase, where in this example, this individual in the little cohort, you see quite frequent skipping of this 5th exon, whereas in controls, you almost never see skipping of that exon. And they also found a rare variant near the 3' end of this exon, which is likely the cause of this exon skipping. So that's sort of the rare disease setting. We and others have also used Leafcutter to look for splicing quantitative trait loci, so genetic variants that seem to be associated with splicing patterns. This paper from Dan Gaffney's group in the UK used Leafcutter to map splicing QTLs in iPSC-derived sensory neurons from just over 100 individuals. And this is an example of one of the splicing QTLs that they found in this CYP1L2 gene, where the more increased minor allele count was associated with increased inclusion of this exon. And what's interesting in this setting is this variant's also strongly associated with Parkinson's disease risk. We've also looked at using Leafcutter to map splicing QTLs and compared to existing approaches. And we find considerably more splicing QTLs. And we think this is a combination of both being very flexible in terms of the type of splicing events we can represent, and compared to the transcript ratio approach, having much lower noise. Something else we've used Leafcutter for in a similar vein is an analysis called a transcriptome-wide association study, or a TWAS. So the idea of a TWAS is that if you have some large case control cohort where you have genotypic data for the individuals and also some trait of interest, the example I'll show you will be late onset Parkinson's disease, then what you traditionally do with this sort of data set is to run a genome-wide association study, or a GWAS, which tries to directly associate genetic variants, typically single nucleotide polymorphisms, with the trait that you're looking at. Even if you do have enough statistical power to find these associations, there's still a challenge in that you have no idea what the molecular mechanism is linking these variants to the trait. And one of the big surprises of GWAS is that the majority of findings, around 90% for most phenotypes, have actually been found in non-coding regions, so they're not affecting protein sequence. What a TWAS does is to instead leverage external reference cohort where you have genotypic data and RNA sequencing, so for example, GTEx, to build models that predict gene expression from local genetic variants, and then use those models to predict what the gene expression levels would be in the GWAS, or case control cohort. You can then associate those predicted expression levels with your trait of interest. Something that's appealing about this approach is that compared to if you had associated observed gene expression with the trait, you have less worry about reverse causation. So if you do that latter approach, you have this concern that the associations that you would see between disease and gene expression, they might actually be effects on gene expression that are downstream of the disease. Whereas here, because all you're capturing in these predictive models is the genetic component of gene expression, you don't have to worry about that as much. So while this has been done previously for expression, the leaf cutter quantifications let us also do this for splicing variation. So we've done this, for example, using publicly available data for Parkinson's disease. And interestingly, we actually find more genes associated through splicing variation than through gene expression. So what this plot is showing is z-scores. So here, higher absolute value means more significant. Green points correspond to association through splicing, and the blue points correspond to association through total expression, and then black points mean that there's both. And the strongest association you see here is for MAPT, which is microtubule-associated protein tau, so exactly the gene I showed you earlier. However, our data makes it look like it's, in this case at least, it's not exon 10, which I told you about earlier, but exon 3 that seems to be driving this association. So this variant in the intron between exon 1 and 2 is strongly associated with Parkinson's disease risk, and actually the minor allele is protective. And something that's sort of fascinating about the TWAS approach is not only does it give you a hypothesized molecular mechanism for that association, it also gives you a direction of effect. So here, the data would suggest that increased inclusion of exon 3 is actually protective against Parkinson's disease. Of course, you need functional follow-up to really be convinced of this. So both the splicing QTL analyses and the TWAS approach are all about understanding the effects of variants that we've seen before in human populations on splicing using association testing in different ways. But there's actually a lot of times we'd like to be able to predict the effects of variants we've either very rarely seen or never seen before in a human population. And so is there anything we can do there? So one approach would be to try to understand from first principles how genetic variants might influence the splicing process. The splicing is an incredibly complicated biological process involving hundreds of proteins and non-coding RNAs. I'm showing a tiny subset of those here, along with some of the splicing motifs that are known to be important for determining splicing choices. So because understanding this process in detail is so challenging, we've taken a more data-driven approach. So our idea is to try and predict splicing outcomes directly from sequence, and then query our models for what effect the model predicts a change in that sequence would have. So the way this would work is to consider a fixed anchor, what I'm going to call an anchor intron start, so a 5' splice site, and then try to predict where the end of the, however you want to view this, the end of the intron would be, or the end of the junction, or the corresponding 3' splice site. That will almost always correspond to an AG dinucleotide, and while I'm showing these at known exons, apart from this one, an AG will occur on average every 32 nucleotides, assuming random sequence. So there's a lot of these AGs. What we do is to take an 800 base pair window around each of these AGs, feed that sequence into a neural network, which for us is just a nonlinear regression, which tries to predict where splicing will occur. And we can then fit these neural nets to try and match what we observe in RNA sequencing data. And in all the examples I'll show you, this will be from GTEx brain RNA-seq. Yeah, and we use, the quantification we're using is therefore very similar to what we're doing with leafcutter, and we also use the same likelihood model, so the Dirichlet multinomial. The neural network that we use is designed to account for the sort of expected splicing biology. So in particular, we expect that there are specific base pairs around the AG that are very important for determining the strength of the splice site. And then there are more distal effects where through splicing regulatory elements, where the exact position of those sequence motifs, some variation is allowed in that positioning. So for the former, we use a dense neural net, whereas for the latter, we use a convolutional neural net. And those two signals are combined through two more layers of neural network to give a final prediction of the splice site strength. So the way we've assessed performance with this method is to look at prediction of splice site usage on held-out chromosomes. So this is training on odd-numbered chromosomes and testing on even-numbered. The metric I'm going to use here is the proportion of the time we correctly predict the most frequently used 3' splice site. And remember that I said that AG is going to occur on average every 32 nucleotides. And something I didn't say is we're always looking 100 kilobases downstream of that 5' splice site. So that means on average, typically there's going to be on the order of sort of 3,000 AGs. So if you guess at random which AG is going to be the most frequently used, you'd get the right answer only 1 in a 3,000th of the time. So the fact that this work from Chris Burge's group, and this is Jean Yao, from 2004 gets the answer right about 10% of the time is actually pretty impressive. Then the best dense neural network we could come up with gets the right answer about 20% of the time. The best convolutional neural net about 30% of the time. Our method, Grasshopper, gets the right answer about 70% of the time. If you don't include overdispersion, so this is if you use a multinomial likelihood instead of that Dirichlet multinomial likelihood, then you lose some performance. We also include intron length as a feature, and it turns out that that's also really important. If you don't use it, you lose a lot of accuracy. Something else that's interesting is if you include a phylo-P conservation track, so this is just saying how conserved is each base pair across mammals, then you can do quite a bit better, which clearly means that there's signal that we're still not able to find in the sequence, because the spliceosome clearly does not get to look at a phylo-P conservation track. We don't use this in our later experiments on the basis that it's not sort of giving a mechanistic view, any additional mechanistic information about what the spliceosome is doing. So we can then use in silico mutagenesis to do several things. So in silico mutagenesis is just this idea that you would change nucleotides in the input sequence to the neural net and ask what the change on predicted splice site strength would be. So this is doing that for every nucleotide around a specific 3' splice site. And so this is for some anchor 5' splice site that's way off upstream somewhere. And what's amazing about this is that from scratch, the model is able to recapitulate many of the known core splicing motifs, for example, the branch point sequence, polypyridamine tract. It also learns that exons tend to be GC rich. And it also, so this is just for predicting whether this would be a 3' splice site. And it learns that to be a strong 3' splice site, there needs to be a 5' splice site consensus sequence a little downstream as well. So this was just for one specific splice site, but we've also done this across all of the test splice sites and done k-mer analyses to try and understand genome-wide what signal is the model using. So this finds several of the signals that I mentioned earlier, so consensus branch point, 5-mer, polypyridamine tract. The red here corresponds to the consensus 5' splice site. But interestingly, this also pulls out one of the largest classes of exonic splicing enhancers. That's sort of an additional signal beyond the core splicing motifs that also encourages splicing inclusion of the exon. And these have previously been looked at in various papers from Chris Berger's group. We can also use in silico mutagenesis to predict effects of variants that we actually observe in individuals. So this is doing that in an example of doing that in GTEx. So this variant, which exists at very low minor allele frequency in Europeans, changes this G to a T, one base pair upstream of this AG. And while this is used very rarely as a 3' splice site in all the individuals who have a G in this position, so who are homozygous major allele for this variant, the one heterozygote individual in this, in GTEx, you see almost exactly 50% of the RNA-seq reads aligning using this junction, suggesting that whenever you have that variant, that junction is used, and as the model predicts. So more interestingly, we've applied this to large sequencing cohorts, so particularly for intellectual disability and also for autism spectrum disorders using including the Simon's simplex collection. And the controls we used were also from Simon's. So our approach here is to use Grasshopper to categorize intronic and synonymous variants as whether they would be predicted splice disrupting or not, and then compare the number of those mutations in cases to controls while trying to account for different de novo mutation ascertainment rates because of different coverage across samples, and also mutation rate being dependent on local sequence context. So when we do this, we get an estimate of that the individuals with intellectual disability have just over 1.5 times more predicted splice disrupting de novo mutations than controls, and that number for autism spectrum disorders is just under 1.4. Both of those enrichments are statistically significant, and by running a parallel analysis where we look at protein truncating variants, we estimate that around 7 to 16% of pathogenic de novo mutations in these settings would be likely acting through splicing. We can also add these predicted splice disrupting variants to burden tests that try to highlight genes likely involved in the disease, and we're able to find a small number of additional genes through that approach. Those effects that we're using are all predicted effects on splicing. So we've also done RNA-seq, probably confirmation would be a better word than validation here, in lymphoblastoid cell lines, these blood-derived cell lines from Simon's. One example that we found was a predicted splice disrupting event in this MED13 gene, which is involved in the mediator complex, which is broadly involved in transcription. So this de novo mutation changes this A to a G just downstream of this exon, and this GT actually ends up getting used as the 5' splice site rather than the canonical GT. And what that seems to result in is quite frequent also use of this GT as the splice, as the 5' splice site, but also quite frequent skipping of the exon entirely. And obviously, since this is a shift of 2 base pairs, this results in a frame shift. Overall, we see about 75% of the predicted splicing dysregulation events validating. The false positives that we have here it's likely, we think, that at least some proportion of those are as a result of the predictions we were making were for brain, whereas the RNA sequencing we're doing here is in this weird blood-derived cell type. So in terms of future directions for the lab, the goal that we have is to try and understand more deeply RNA splicing's role in human genetic disease. So I see two aspects to that. The first is deeper understanding of how genetic variation can change splicing choices, both for common and rare variation. There's various approaches to that. The grasshopper model does a good job of predicting splicing outcomes for, on average, across tissues. But where we see differences in splicing regulation between tissues, picking up on those signals is very challenging. So that's one direction that we'd like to improve on. We'd also like to have approaches that scale to looking at splicing QTLs in really large cohorts. TopMed is hopefully getting up to 15,000 RNA sequencing samples at some point. And also leveraging massively parallel reporter assay systems. So there's been nice work using degenerate k-mers to look at, in an unbiased way, the effects of changes in sequence on splicing outcomes. But then there's also, even if we observe these effects of genetic variation on splicing, there's also a question of whether confirming that those differences are actually driving disease. So we're interested in looking at that in a number of settings, in particular leveraging RNA sequencing data that's now being collected here in the context of autism. So really leveraging the RNA sequencing as a part of the discovery process rather than just as validation of our predictions. In collaboration with many people here working on ALS, we are interested in looking at functional validation in patient-derived, iPSC-derived motor neurons from patients. And then something that we're excited to work on with Naval Sanjana's group here is trying to adapt CRISPR-Cas9 forward screens to Cas13 screens, which would be RNA targeting rather than DNA targeting. So being able to, in a high throughput way, recapitulate the missplicing that we see in various disease settings and try to understand which of those events are likely causal for some phenotype. I'd like to acknowledge many people who are involved with this research along the way, and very happy to take questions.
Thank you so much. Great talk. I have a question about the transcriptome-wide association.
Yeah.
In the first part, you identify SNPs that is associated with transcription changes.
Yeah.
So how are you? I just wonder how different it is from the EQT identification in terms of methods.
Yeah, I mean the idea is very similar. The main difference is usually when people are doing, not always, but usually when people are doing EQTL studies, they're thinking about the effect of one genetic variant at a time on expression or on splicing. Whereas, and they're not interested in prediction. So then you don't need to think about the effects of multiple variants at once. The type of models that people standard for TWAS are things of regularized linear regression models is what's usually used. So lasso or elastic net is what's used the most. So there you're learning a joint model that's trying to, that will actually typically include a lot more genetic variants, SNPs, than you would call as significant in a EQTL study. Because you don't care so much about trying to say like which one might be causal or how many genetic variants are there here. You just care how well can you predict gene expression based on the local cis regulatory landscape. Yeah. So slightly different aims, I think, is the difference mainly. Yeah.
Can you talk more about the training data that you used in your splice site prediction, and whether expanding the training sets across individuals or across tissues, is that a small effect, or will that be transformational?
Yeah, so actually the fact that the result I showed showing that when you don't include overdispersion, that the performance drops a little bit is actually related to that question. So if you don't include overdispersion, the multinomial model means it's the same as if you just concatenated all of the RNA-seq data you had from all of the individuals as though that all came from one individual. So you are gaining a little bit in terms of having multiple individuals, but it's sort of just denoising. Because really the sample size for that training is the number of splice sites you get to see. And okay, you might get, because of, if you go to more individuals, you get more coverage. If you go to more tissues, more genes are expressed. So you do get more training data from that point of view. But like doubling the number of individuals you have is definitely not doubling the number of splice sites that you get to look at. So actually, I think for the thing I was saying about, we'd love to have better tissue-specific predictors to get more sample size there. I think actually you need to go to look at data from other mammals. We think that the splicing machinery is relatively conserved for the most part across mammals. Which kind of goes back to the point I was making about the conservation of splicing differences that we see between tissues across species. And the other way to go is the massively parallel reporter assays. So then you can obviously potentially generate a lot more sequence to train on and not just be limited by what you see in natural variation. Yeah. That's great.
So this is wonderful work. I wanted to ask, have you looked, you talk mostly about mammals, but we have this wonderful history of molecular genetics, especially like in flies and these model systems. If you've gone to find new biology in, double sex and sex lethal and like all these things that we discovered and then sequenced, and if you find new biology or at least show like that your process strongly recapitulates what we know.
It would be really, I know we haven't. It would be cool to go back. I mean, there's been so much, absolutely. There's so much done in fly. I mean, there's really cool stuff with DSCAM about like controlling neuronal connectivity and all of the, sex determination stuff. So, it would be cool to go and try to leverage some of that as well. It's not something we've done. There's crazy splicing in plants too, all this like crazy intron retention everywhere, which, actually, and that's a caveat, is we don't cover intron retention very well. You get sort of indirect signatures of intron retention coming up, but we don't sort of directly try to quantify it. So.
But that has been done with double sex. You can use them interchangeably in mammalian cells and flies, and the same binding sites for SR proteins. Everything seems to be really the same.
It's cool.
I have two questions. So the first question is about your first tool, leaf cutter, which you showed an application which was submitted, I think, Nature Communications. So if you can comment a bit how using leaf cutter would compare with just if the authors were just using one of the tools which look at de novo variants and predict pathogenicity, and probably they would have found those variants just looking at this.
I don't know. I mean, it's
what would your tool add an extra of just looking all the novo candidates, doing some prioritization and
I mean, these are all the individuals they were looking at there were individuals who'd had exome sequencing already and hadn't come up with any diagnosis there. So I think they would have looked, when they just had the exome sequencing, they would have looked at predicted pathogenicity of all of the variants they were finding there. I mean, I think you just have a lot more, it's just a much richer information source once you have the RNA sequencing to leverage, right? Because you're seeing the things that are closer to phenotype you know, there's going to be an enormous number of variants that could be causing, not an enormous number of de novo mutations, but an enormous number of rare variants that could be having effects.
But were those they have found with your tool de novo, or?
The example I was showing was, that was a rare variant, not de novo. They didn't have families here, so they don't know. I think they actually don't, I guess they don't know what necessarily whether things are de novo, but at least that was a known rare variant. It's not like for all of those they know, it's not like for all of those that things where they found what they're saying as a diagnosis, they necessarily have a genetic variant that they think is driving the gene misregulation.
You mean with your tool? So in some cases it was not a genetic variant.
Or they don't, well, I mean, they don't know that it would be or they, likely it is a genetic variant, but they don't know what genetic variant.
Okay. The second question is about the neural network. So you said that you're using these flanking regions, but you also added phylopy. So how you change the input to the neural network when you add conservation, and conservation of all the flanking regions positions, or which ones?
Yeah, so all of them. So the actual input to the model for the convolutional part is we do what everyone does, which is a one-hot encoding. So effectively, the analogy to, so the convolutional neural networks primarily come from computer vision, and then you're looking at images, so you have a width and a height of the images and some number of channels if it's normal imaging, RGB. So the one-hot encoding that you use if you're trying to run a neural network on sequence means that you have 4 channels. So instead of RGB, now 3, you have 4, which just correspond to the 4 nucleotides. And then instead of it being an image where I have 2 dimensions in the input, I just have 1 dimension because I just have where in the sequence am I. So if you have any input which is per base pair, then you can just concatenate it onto that. So if I include conservation, I would just add in, now I have 5 channels. So I have the 4 channels encoding which nucleotide is it, and a 5th channel which is phylopy conservation. If you look at a phylopy conservation track, the strongest signal is that exons are much more conserved than introns. So that's clearly like some of the signal that it's looking at. People have done similar things with predicting transcription factor binding, where you'd have the one-hot encoding, and then the per position would be chromatin accessibility measurements. So things like ATAC-seq or DNA's hypersensitivity. Yeah. Yeah, we've done some comparison with the spider stuff.
Might be more recent.
Okay, I don't know if we looked compared to. I don't know if we looked compared to that one. I don't think we haven't compared to that one. Yeah, they're different setups in that the Brendan Fry stuff is basically always trying to predict exon inclusion levels and not trying to do this question of predicting where the actual junctions will be. So then maybe that's the equivalent thing. Okay. Do you know when is it published or it's on bioRxiv? Or? Okay, I don't think I've seen this. Okay. Okay, I don't think I've seen it.
I guess it's the bigger picture question, but do you see any sort of, beyond the obvious parallels between like the systems biology work and the machine learning work? Both, have evolved. One's evolved amazing species, and the other one is finding very hard to find solutions that we can't do with, less complex mathematics?
So I don't know if I'm interpreting the question correctly. So you're saying like the AlphaGos of the world that needed very complex mathematics, is that what you're saying? And then
just sort of like, there's genetic models for doing stuff, and they have that name because they borrow some ideas from biology, and yet we're like using similar sort of systems of mathematics that maybe, I don't know, is found in nature. And
Yeah.
I don't know, more philosophical.
Yeah, it sounds like a very philosophical question. Yeah, I don't know where to go with that.
Okay, no problem.
So most of the examples you gave were creation of new splice sites. Have you tried to look for sites within exons that do not affect the splicing recognition, but affect the SR protein binding sort of enhancement of splicing itself?
Yeah, those signals are definitely weaker. And I mean, you saw, right, we ran this on all of Simon's, and then we ended up following up on only 28. I don't think any of those were actually that sort of more subtle modulation. That signal is definitely weaker, and the predictions are not as reliable compared to the splice site creating disrupting variants. Yeah, we'd love to be able to do a better job at that. Yeah.
Any other questions? If not, I'd like to thank you for a great talk.